So we have a couple questions. I'll start up at the top. We have a question from our known dog. This is for you, Patrick. Can you please refer, what is the effect of biostimulants on plant hormones?
Yeah, Anon, nice to hear you all the way from Israel. Yeah, it's an interesting area. Originally, the seaweed guys and also the humic guys did some analyses of the hormonal content. They were trying to reasonably explain what's going on. And certainly these products have small amounts of hormone in them, though the analysis shows it's really, really very small. And that's not surprising. Everything vegetative has hormones in it. In fact, a bowl of spinach has more hormones in it than a bowl of seaweed, to be honest. They have, however, backed away from making those claims, and it comes back to the regulatory framework. If a biostimulant claims to have hormone or hormonal-like activity, then it risks being classified as a pesticide. And you would then have to go through all of the EPA residue testing, human testing, and it would be hugely expensive and impractical. So it's a sort of and I have this discussion, I am having this discussion next week with USDA and EPA as to whether that is doing us good or harm. Have that clear boundary, because it's compromising our ability to really say what's happening.
Great. So the next question,
in case they find hormones. So that's part of the discussion. And as I said earlier on, the reason many of these companies are calling them nutrient enhancers or fertilizer-like products is exactly to avoid the pesticide label. So long story short, I think they do interact with hormonal pathways. But then again, any event in a plant, a stress response, a water stress response, a nutrient response, a plant growth response, they all trigger hormones. Of course they do. That's how growth occurs. So we're dancing that dance right now to try and work out how to deal with this.
Great. So the next question is, the example in Midwest Prairie is one of organic plant growth. What impact, if any, does the application of chemical pesticides such as Agriment for thrips or glyphosate for weed control, or even Arindus for root rot protection have in regards to the effectiveness of the biostimulants in the plant microbiome?
Yeah, I mean, it's difficult to answer. We don't have enough knowledge of which soil microbial component is the important one. So while it's easy enough to demonstrate these products alter the microbiology of the soil, it's harder to demonstrate that it matters. And then another example, and I put this in the chat as well, when we think back to strawberry production, methyl bromide kills everything. You have essentially a sterile soil. And yet in the context of strawberry production, it's profoundly effective. And that might simply be because in strawberry production, you manage the water, the nutrients, and the pathogens to such an extraordinary extent that you don't need the soil microbes to help you out. If you were growing in a situation where you're not managing all of those inputs and controlling them to such an extreme extent, then it would be rational to say adding these microbial pesticides would have a negative.
We lost you here for a second, Patrick.
Was I talking to myself?
Yeah, a little bit, but I think we got the message. So you also answered the question in the chat to Enrico that asks, we need to know the ideal microbiome for our particular crop to know what the deficiencies are, and is there any way to measure the different aspects of a plant microbiome in non-native environments? I mean, I think when we think about the native habitat of avocados, they're from the temperate zone, subtropic rainforests.
Sorry, my sound dropped out until the last 3 words. So quick.
Oh no. Okay, so I read Enrico's question. Wouldn't we need to know the ideal microbiome for a particular crop to know where the deficiencies are? Is there a way to measure the different aspects of a plant microbiome in non-native environments?
So we're not there yet, and the current sort of dogma on soil health from USDA and other locations is the more diversity, the better, and the bacteria and the fungi will work it out. That's a pretty crude statement, but that's as good as we've got because the complexity is not understood. There's a couple of companies that offer soil microbe tests, and they will categorize how many of various functional groups you have in the soil. And I think they do an analysis that simply says, we went to a great orchard and it had this profile. We went to a terrible orchard, it had this profile. Therefore, the great orchard profile is what you want.
We lost you again. Sorry.
In the next decade, the progress in this area is absolutely extraordinary.
Okay, we have a question from a grower who I think wants to know how many trees or how many per treatment are required to provide potential statistical analysis. So, ideally, if you go back to the old publications of Embleton and Jones and Labanauskas, they suggest 20 trees for avocados because they're so variable, even on clonal rootstock. They recommend 20 trees. I think if you have a uniform site, the soil is uniform, the slope is uniform, et cetera, you might be able to get that down to 10 to 15 trees, but I would never do less than 10 trees. And as Gary said, it's very, very important to have an untreated control so that you have something to compare to and make sure that they're sort of grouped in comparable situations so that, for instance, the control trees are not downhill in a cold spot in a grove and the treated trees are uphill in a better situation. Miriam, your iPad is on. Miriam, if you could, thank you. If you can mute your, thanks.
Mary Lou.
Gary, do you have anything else to add?
I wanted to say that one slide I had with the, where it had Grow Getter at the top and then it had the non-treated control at the bottom. I think the non-treated control had like 41 fruit on it or something like that. The water sprayed trees had 73. And I showed that data at a meeting one time and a grower said, look what just the water did. So he actually, next spring, he went out with his helicopter and sprayed a whole bunch of acres. I think he did 40 acres of water. And I said, that was only 1 year of data, and we repeated that and repeated that. And then sometimes the water spray was just exactly the same as the non-treated. So you can't really go by that. And I said, where's your control? He didn't have a control.
Maybe he was controlling the mites, hey, Gary?
Good point, Patrick. I don't know, but he was spraying right in the middle of the flower season when I was spraying. So maybe it just cooled them off for a day or something.
It could be, that could be.
And one of the things with regard to testing, and we've not done a lot in avocados, but I've done pistachio, which is almost as problematic, is we like to take a lot of individual data from each tree. So rather than doing blocks of 5 trees and taking the average of that block of 5 and calling it a replicate, it's almost always better to treat the individual tree and have individual tree replicates, but also collect things like trunk circumference and leaf area and things like that so that you can normalize some of the differences between trees.
Yeah, I agree.
Gary,
really
Dave Holden
important.
here. Can I jump in on that?
Sure, Dave.
I remember, and we all remember who did that. And their theory, if you remember, was that if you spray the trees with water, the pollen will stick better and you'll get better production, if I remember correctly. Was that right, Gary? That was like 25 years ago.
It's so long ago, I don't remember what the theory was on that. I'm sorry, I just don't remember.
It just goes to show you, when we say something as experts, we got to be careful what we say because it's taken to heart and people start doing it.
Yes, it is.
Hey,
yes, it is.
Thank you to both of you. Excellent presentation for both of you. And I needed it. Thank you.
Great. We have a question here. Has anyone used Revitalize by Bonide? It's a bacillus, and I won't even, liquefaction strain D747. Patrick, do you know about that product?
I've not used it. That Bayer product that was very successful was a mix of a bacteria amylofaciens and liquefactions and amylofaciens and subtilis, and it worked. But you know, the strains that are available, there are thousands and thousands of them, and each company claims theirs better than the other. There is something to do. There's something to say about those bacterial microorganism biostimulants, but they're not any more predictable and reliable than the other biostimulants. Sometimes great, most times not.
How do you apply them, Patrick?
The one I applied on behalf of Bayer was an injection into the drip irrigation system.
In avocados, you know, we have heavy leaf mulch. You can have it, that mulch can be 6 to 8 inches thick.
Yeah.
And a lot of people will have sprinklers rather than drip. Would you have to worry about that bacteria getting down actually into past the leaf mulch?
That's a good question. Yeah, you'd have to test that.
So we have a question on, do you have any thoughts on humic acid?
I know Dave probably does, but you know, his customers probably don't want him to talk about it. Originally, you know, humic acids are interesting. Early on, they sold themselves as contributing cation exchange capacity to the soil. And then if you do any calculations of how much percent organic matter is in the soil and what's the humic fulvic content of natural soil organic matter, you soon realize that adding a liter or 10 liters of humic acid could not possibly alter the cation exchange capacity. Having said that, subsequent work in Spain sort of indicates that what the humic acids may be doing is interacting with the root membrane and triggering a sort of a stress response in the root. So it sort of triggers the plant to perceive a stress is present. And depending on when that timing is, it can be beneficial. So there were some benefits shown from that. Humic acids suffer from, not all humic acids are the same. Whatever resource they're mined from in North Dakota or West Virginia or wherever else, that makes a difference. How they're extracted, alkaline extracts, whether they go through a molecular sieve or things like that. That's the problem, is you can't really say all humic acids are the same. It's like saying all composts are the same. So they fall into the, yes, there is some evidence, and there are a lot of growers who swear by them. And I trust growers over time to have worked out what works and what does not work. Are they explained? No.
Yeah, well, you know, that's interesting. This is many years ago. I went to a grove over in Bonzo. They had sprayed with humic acid. Don't ask me which one. But this was an older Hass grove. But those stems on that avocado, you could barely get a clipper around the stems. And it was amazing. And I mean, it had, I believe it had to do with the humic acid. They had very high yields in that grove, but the stem diameter, I remember on that fruit, it was almost impossible to clip the fruit.
Now, was that applied through the irrigation system or sprayed?
I don't, you know, Ben, I mean, Gary, I don't remember, but I mean, I remember that was associated with they used humic acid in that grove. And then when they stopped using it, we went back and the stems were thin. So I don't know. But
yeah, and I think a number of growers have seen that. And I've been in orchards where it's like, wow, what'd you do in that half of the field? And they're bigger and greener and thicker and they look nice. You got to be a little bit careful. In the early days of biostimulants, people were throwing urea and iron and zinc in with their humic acids and urea, and it would have a greening effect, and they'd call that success. But it might have been the urea and the zinc, and the iron, not the biostimulant itself.
so we have a long comment, but I think it's worthwhile to read that it's addressed to you, Patrick. In Spain, private laboratories are starting to implement relatively cheap metagenomics analysis for fungi, bacteria, virus, viroid plasmids. In the rhizosphere zone, this would at least let us know what families or general species we may have and in what relative numbers. It would be one way to measure variability. Another thing would be to know what these microorganisms do, fix nitrogen, solubilize phosphorus or potassium, emit cyanophores, etc. This could be known through respiration analysis, metabolic activity. But I share a lot of what is being said. What is a healthy soil? How much do I have to help the microbiota to with the inoculation, and do they really have an effect on the crop with diseases, or so that I can reduce fertilizing the trees by half without losing production? And how can I measure this?
Yes,
I mean,
that's where the-
basically, how do you do, I mean, how really do you do that kind of research?
Great question.
It is a great question. It's sort of asked and answered. Yeah, I'm familiar with that lab in Spain, and they've had some successes, but I think all of this suffers from us not yet knowing what ideal it looks like.
Yeah, well, it's a further comment. I think I can think of an analysis before the application and after the external inoculation, a follow-up of the microbiome every 2 to 3 months during the campaign. So what do you think? I mean, I remember John Mengi, Gary, remember he was putting out his biologicals, but he couldn't get them to, I remember him talking about they didn't survive.
Right. He said there's so much competition in the soil from other bacteria and fungi, and they're all putting out antibiotics, and they're just not letting the new guy come in. Is basically what he said. So he thought he did some trials with, I think he added straw. He wanted to give them a food base to increase their population, but that still didn't work. You know, he just that was his conclusion. There's just too much competition in the soil already, and that is a difficult thing.
I'm planning to start up a study, I hope, with Johan Laveau at Davis, who does a lot of work on phylosphere microbiota, and it's based on the principle that was coming from human research that says you can give a particular substrate to a malnourished child that encourages a particular suite of bacteria and fungi in the gut. And that is more effective to solve malnutrition than simply giving them a protein bar. Okay, so the implication is the choice of substrate can be used to manipulate the microbiome favorably. And so we're looking at that in the context of the phylosphere and its ability to regulate leaf surface fungal infections by choosing a substrate that favors a bacterial profile that compromises the ability of the fungus to infect. So there's a lot of interest in this sort of targeted metabolic manipulation, but it's early days, and you know we're fumbling around in the dark. There is no question.
Yeah, I would imagine with varying soil pH and different soil composition and whatnot, it would be a very difficult task to be 100% sure that if you were putting material into the soil, that it would even survive to do what you were hoping it would do.
Right. I've had growers ask me what they can do, you know, to manipulate their soil, and they, you know, usually come down to compost or wood chips or something along that line. And a lot of our organic growers do that. And I've looked at organic production from organic grows versus conventional grows, and we haven't really done a study on this, but I just noticed that we seem to have less yield in the organic grows. Now that gets complicated because they're also putting, a lot of them put chicken manure or something like that on as a fertilizer, and a lot of that stuff is high in salt, so maybe they're adding too much salt to their ground or whatever. So it gets very complicated. That's the growers would like to put something on that could, you know, increase their biological stuff in the soil. That's the one thing they can do. But we got to get, if it does work, we got to get the right type of mixture. We do see in some of the grows that when they get root rot, a lot of the grows, just the root rot sort of sits there, moves a little bit, very slowly through the grove. But in other situations, I've seen root rot just race across the grove very quickly and wipe out the whole grove on a hillside. And there's something, those soils, there's something missing in that soil. And most of our soils here in San Diego are on decomposed granite. So they don't have much organic. It's certainly different from the rainforest down in Guatemala. Anyway, that's
So
all.
Gary
Ben-
has his hand up.
Yeah, so
I mean,
Ben,
Ben.
what do you have to say?
Yeah, I was involved with the trial with John Mengi where we had mulch and no mulch. This is in an orchard that had root rot, mulch, no mulch. And then every month I went out and introduced one of these mixtures of bacteria, fungi, trichoderma and Putida and stuff like that. And wherever we introduced this soup, you had to have the mulch there to get it established. Wherever we introduced it with no mulch, it would never get established. But the interesting thing was wherever there was mulch, we didn't introduce it, it came in naturally. So it was like you didn't have to, you had to create that environment that would support healthy bacteria. Well, the point's being made, what is healthy? But we were trying to introduce stuff that would be healthy, and until you created a high organic matter content environment, the naturally occurring good things that we considered good things didn't show up. We couldn't get them established.
And one of the things you got to be a bit careful of, and I showed that slide of the root with all its bacterial interactions. Those interactions occur in the millimeter or the centimeter at the root surface. And they will be quite different in the bulk soil. So when you take your samples to estimate microbial populations, if you don't take it from the rhizosphere soil, it's not entirely relevant. And that's extraordinarily difficult to do. And our ability to characterize and culture rhizosphere bacteria and fungi is less than our ability to cultivate and culture and characterize bulk soil microbiology. So we have another challenge there that complicates life greatly.
That's great. Thank Ben for that comment. So we have another question about humic and fulvic acid. And will that, you sort of touched upon it, but is it known whether if you apply these materials, it'll increase the microbial activity in the soil?
It's mixed. There certainly are some that say yes and others that say we didn't see anything.
Okay, so we have another question. This was from Ben. So how does 12 ounces per acre of some material of bacteria overwhelm the effect of trillions and trillions and trichillions of bacteria that are already in the biome, in the soil?
Very good question. And early on, that would be the retort from any soil microbiologist is there's no way this could happen and this could compete one against the other. And certainly in many cases, it's the case. But there is also, I could point you to 80 papers where they say, no, my small application made a difference. And it's certainly the rationale that the folks like Pivot Bio or Join, who are working with nitrogen fixation in corn crops, they do seed coats. And you can imagine how small that contribution of a seed-coated bacteria is to 1,000 acres of Midwest corn soil at an organic matter of 5 or 6%. And yet they demonstrate persistence. So it's hard to be, it sounds that like you'd never compete with those trichillions, but on occasions you can.
So we have another question. Do you have any thoughts about Acadian algae extract?
Well, that was what I was talking about, actually, that Acadian seaweed from Maine.
Okay.
Yeah, the
And
only thing, I mean, we don't like to make comments of particular product names because their lawyers will call me in the morning. But there are companies, and I think Acadian fits into it, I can say safely, who take their science and their practice seriously. If you look up research in the realm of seaweeds, you'll find 50% of it derived from Acadian. So I give them credit for taking this seriously and doing the best they can.
And then sort of following on that, what do you think about the work of Elaine Ingram from, I think, British Columbia regarding the use of compost teas?
Yeah, I'm not familiar with it.
Yeah, I know if you go to South Africa and you go and visit the very large farming operations, too, they're big believers of compost tea. And in fact, within their own farming operation, they have like a sub-industry of making compost teas and applying biologicals. And they're big believers that they've made a difference. They grow avocados, greenhouse tomatoes, and a lot of onions. And they claim that it really, really works on the tomatoes. But I don't know. They have a mix of different things that they apply.
Yep. Yeah. Last couple of years, we've done work with returning almond hulls and shells back to the almond orchard. It was driven primarily because 80% of the orchard's potassium was in those hulls. So why are you paying for fertilizer and paying for haulage? And they were quite remarkable, not only in supplying potassium, but other nutrients and in improving soil penetration, soil health characteristics, water retention, and water characteristics. Now, that's not compost tea, but it's sort of the concept that you put back to the system what you took away from the system, and you end up with a balance. So that, there's something to be said for that. It depends probably on how you make a compost tea too.
So we have a question for Gary. Did you ever test the idea of spraying honey diluted with water on avocado bloom to attract bees and increase fruit set? But I think if our known dog is still on the call, that would be a great question for our known to answer, since he's worked on bees.
Well, it's easy for me to answer. No, I haven't done that.
Okay, our known.
There are many compounds that people try to attract the bee, but if you are looking in the literature, it's a little bit similar to the biostimulant, that many companies try to market different compound. But if you look for something that show consistent positive response, you will not find anything. You cannot cheat the bees. Even if if you'll spray with honey, they will visit the leaves, so it won't help you. You need the bee to visit your flower, so I don't think it will work.
Thanks, Arnon. Do we have any other questions? We're right at 10 o'clock. I don't want to cut it off if there's more questions that people want to ask. I don't see any more questions. Anybody want to raise their hand and make any more comments? If not, Arnon, thank you for your comment. And I really want to thank Patrick and Gary for participating. I apologize for that we can blame the rains, the very, very much needed rain and snow we've received in California, but it's caused havoc, I know, here in the San Joaquin Valley on infrastructure. So I hope you can be patient with what went on today with the audio, but I think the message came across loud and clear. And I want to really thank Patrick and Gary for talking on today's Avocado Cafe. So thank you everybody for participating. And we are putting a meeting together for probably sometime the end of February or very beginning of March, and you will be getting a notice about that. And please, if you are in California or you're interested in listening to the Arindis presentations on the 31st of January, please let either myself or John Cornell or Johannes or Avocado Cafe know, and we'll make sure that you get that link. Thank you very much and have a wonderful day.
Thank you.